Eukaryotic cell division involves mitosis, which is going to be the nuclear division, and cytokinesis, which is division of the cytoplasm. During mitosis, sister chromatids are separated and distributed into daughter cells. So before mitosis can begin, the chromatin is going to have to condense down into distinctly visible structures. And each species is going to have a very characteristic number of chromosomes. Chromatin. So we've already kind of talked about how DNA in eukaryotes is associated with the presence of certain proteins called histones. When a eukaryotic cell is not undergoing cellular division, the DNA and associated proteins are located in kind of a tangled mass of very thin threads. Before mitosis can begin, the chromatin has to be condensed down and coiled into tight structures known as chromosomes. Because these structures are so tight and dense, they can actually be seen with a light microscope, and we should be able to see them in our mitosis and meiosis labs. When these chromosomes are visible like this, they're relatively easy to photograph and count them, and each species, as we mentioned, has a very specific number of chromosomes. Here's just a table showing some of the diploid numbers of chromosomes for various species. The number listed here is the full number of chromosomes found in the cells of individuals. This is what's also known as the diploid number. The diploid number includes 2 chromosomes of each type of chromosome. So for example, humans have 23 different types of chromosomes, but every single cell in your body has 2 copies of every type of chromosome because you get 1 set of DNA from your mom. So you get 23 chromosomes from your mom and you get 23 chromosomes from your dad. And so our total diploid number is 46. Yeast are the same way. Yeast have an even set of chromosomes. They have 32 chromosomes, but they have, 16 individual types of chromosomes, 16 different types of chromosomes with a total diploid number of 32. Just because you have a certain amount of chromosomes doesn't mean that the individual is necessarily more or less complex. For example, we have 46 chromosomes and the fruit fly only has 8 chromosomes. That seems fairly straightforward. However, the goldfish has 94 chromosomes. Goldfish have almost double the amount of chromosomes that we have. I'm sorry, not almost. They have more than double the number of chromosomes that we have. And the southern addern's tongue fern is a plant. It is a fern that has over 1,000 chromosomes. So there is a huge number of, discrepancy in the number of chromosomes associated with each organism. So the diploid number indicates 2 sets of chromosomes for each type. Like we said, humans have 23 different types of chromosomes, thus we have 46 chromosomes total. Each type is represented twice in each body cell or each diploid cell. Only the sperm and egg have 1 of each type. So if you're a female, all of your eggs have only 1 of each type of chromosome. There are only 23 chromosomes in each of your egg cells. If you are a male, there are only 23 chromosomes present in each of your sperm cells. Why might that be? All right, I'm going to just leave that out there. But these eggs and sperm cells, these types of cells, are what are known as haploid cells. Haploid meaning that they have half the diploid number. The haploid number for humans is 23. We have 2 representatives of each chromosome type, which makes a total of 2n, which is just 2 times our haploid number. So we have 46 chromosomes inside of every nucleus. 1 set from the individual's father and 1 set from the individual's mother. So we have 23 paternal chromosomes and 23 maternal chromosomes. For now, we're just going to be studying cellular replication and division. We're not talking about sexual replication, so that means that in the case of mitosis and cytokinesis, we are only concerning ourselves with diploid cells and how diploid cells replicate to become more diploid cells. And so the cells that we are studying in this chapter are going from having 46 chromosomes to having 46 chromosomes. This kind of replication is absolutely essential for growth and development. Every one of your skin cells die and slough off. They must be replaced by new skin cells. These cells then are replaced by cellular replication. Sexual replication involves two haploid cells from two individuals, the paternal and maternal DNA, to produce one diploid individual. But like I said, in this chapter, we'll be talking about one diploid parental cell replicating and dividing to form two identical daughter cells. So, going back to mitosis and cytokinesis, the cell must initially prepare for cellular division. To do so, the cell is going to have to replicate. We're going to need to replicate the number of chromosomes as well as the number of organelles. In this case, a diploid cell replicates and divides into two identical cells. The dividing cell is called the parent cell. The resulting cells are going to be what are called daughter cells. So the process of cellular division begins at the end of S phase of interphase. By the end of the S phase, each strand of DNA has replicated. There should now be two identical strands of DNA. These are what are called sister chromatids. They're going to be bound by a single point right here, known as the centromere. Whoops, here we go. This is the centromere. That's going to be the region by which they are connected. This is going to have to dissolve and break apart at some point during the mitotic phase. So during mitosis, here we go. During mitosis, the centromeres that are holding sister chromatids together are going to separate. The two sister chromatids will separate at the centromeres, and in this way, each duplicated chromosome gives rise to two daughter chromosomes. These daughter chromosomes will be distributed to the daughter nuclei of the daughter cells. In other words, we have all of these duplicated chromosomes. We're just going to tear them in half, split them apart, and one copy will go with one cell and one copy will go with the other cell. And we're going to end up with two completely identical daughter cells. Here is our structure of our chromosomes. We have our two sister chromatids bound by the centromere. What we have on either side is a protein structure known as our kinetochore. The kinetochore is it's kind of like a protein handle that's attached to the outsides of those sister chromosomes. Our mitotic spindle fibers are going to reach out and grab onto those handles right there, and they're going to physically pull apart. They're going to retract those spindle fibers and pull those sister chromatids apart right at the centromere. So like we said before, mitosis is a carefully organized and highly regulated process. In order to direct the movement of all of the chromosomes, the cells rely heavily on the help of centrosomes to organize and arrange where everyone is supposed to go. They're kind of like the drum major of the marching band. Here are our centrosomes. They're going to line up at the either end of the field, and they're going to direct the movement of everybody else. They're going to push and pull the chromosomes until they line up down the center of the cell, and then they're going to physically separate them. We're going to go through each one of those phases of mitosis that we have talked about. So starting off with prophase. Prophase is where nuclear division is about to occur. So the first thing that has to happen is our chromatin condenses down. We want to keep our DNA as protected as possible. So we're not going to try and separate it when it's all loosey-goosey like a plate of spaghetti. We want to condense down those noodles as tightly as possible into clear and defined structures that can be easily separated. So the chromatin is going to condense. The nucleolus is going to disappear, and the nuclear envelope is going to start to fragment. We're going to see that nuclear membrane, that nuclear envelope start to slowly dissolve and disappear because we need to expose those chromosomes so we can separate them. The spindle fibers are going to begin to form. That's what are known as these asters over here. These are going to be our centrosomes. They're going to be responsible for organizing the main event, and they're going to start to assemble their spindle fibers. Two centrosomes are going to move away from each other. We started off over here in the single corner, but we're going to have to move to opposite poles of the cell in order to direct this process. During pro-metaphase, preparations for sister chromatid separation becomes visible. So not only have we condensed our chromosomes, but we are getting ready for metaphase. The centromere of each of the chromosomes is going to develop two distinct kinetochores. The kinetochores, like I mentioned before, those are those bright yellow specialized protein complexes. They're going to attach to each of the sister chromatids. They're going to physically connect the sister chromatids with the microtubules. These connect the sister chromatids to the poles at the opposite ends of the cells. This is where the handle that the spindle fibers are going to hold onto to physically pull these sister chromatids apart. Metaphase is next. Metaphase is where we push and we pull and we line up our chromosomes right down the center of the cell. This is what's known as the metaphasial plate, where we're aligning the cells right down the equator or right down the middle of the cell. A metaphase plate is just the plane through which the mother cell will be divided. So we have our mother cell right here. We're going to be splitting it in half right down the center. So the first thing we do is line up all of our DNA right down the center of the cell, and then we get ready for division. At this point, we have the M checkpoint, that mitotic checkpoint. This is going to delay the start of anaphase until the kinetochores are attached properly. I like to think of this as the guys who are running like in a roller coaster ride. And the two guys go down either side of the coaster and they check every single seatbelt to make sure that we are ready to go. And then once we have checked all of the seatbelts, they put their thumbs up and the ride can start. That's what's happening at the M1 or at the M checkpoint. We're checking to make sure that we have properly attached these spindle fibers to the kinetochores before we begin our next stage, which will be the division of those chromatids. The division of those chromatids is also known as anaphase. During anaphase, the centromere is going to dissolve. We're going to release those sister chromatids, and as soon as they separate, they are now called daughter chromosomes. So when they were attached, they were sisters. Now that we have separated, they are now daughters. Yeah, the familial terminology is a little bit wonky. I'm not going to be terribly picky about it in this class. I understand that it's confusing when we're going from parent cells to daughter cells to sister chromatids to daughter chromatids. I understand that it's a little bit wonky. But what I would like you to know is parent cell versus daughter cell. At the point where it's the daughter cell, we have gone from the initial single parent cell, we've divided it in half into two daughter cells. But those two daughter chromosomes are going to be pulled to opposite poles along those kinetochore fibers that are slowly reeling them in. Those poles are going to be moving further and further apart as the cell continues to expand, and we're going to eventually separate that parent cell into two daughter cells. This is going to start to happen during telophase. Telophase, we're going to see a reforming of the new nucleus. So the spindles are going to disappear and new nuclear envelopes are going to form around those daughter chromosomes. Rather than create a nucleus and try and shove the chromosomes inside of it, what we're going to do is just dissolve the nucleus to begin with, and then all that we have left is those all we have left are those sister chromosomes. Then we're going to separate those sister chromosomes and reform a brand spanking new nucleus right around the outside of it. Now we're going to have two clusters of daughter chromosomes. We still have two of each type of all are going to be represented. We have clusters are going to eventually become those daughter nuclei. The nuclear envelope forms around those two daughter nuclei, and each daughter nucleus receives one chromosome of each type. Division of the cytoplasm is going to require cytokinesis. At the point where we have separated our genetic material, we have finished up mitosis, but we still have these cells, the cell is still attached. We still have, cytoplasm flowing back and forth. So now we need to undergo cytokinesis. Cytokinesis is going to pinch off the rest of that cell. So chromosomes will become diffused chromatin once we have fully formed our new nucleuses. Here is an overview. I strongly recommend holding on to this image or referring back to it in your textbook when you start going through your labs. You will be asked to identify several pictures from a microscope, and this diagram will help you identify the stages of the cell cycle.